Welcome back to the deep dive. Today, we're really getting down to the fundamentals.
We're looking at what happens when things go wrong at the most basic level of the cell.
Exactly. We're tracing the journey, you could say, of a healthy cell when it comes under stress, how it gets injured, and, well, how it eventually dies.
And understanding this whole progression of cell injury and death, it's absolutely key. It underpins pretty much every disease we know.
It really is the foundation.
It's the bridge, really. Connects the basic sciences, biochemistry, genetics, all that, to what doctors actually see in patients.
So pathology, that's the term, right?
That's the field. Pathology studies these structural, biochemical, functional changes that are the root of disease. If you get the cell story, you get how diseases work.
Okay, so let's step back a bit. This idea that cells are the key. It wasn't always obvious, was it?
Oh, not at all. For ages, people thought disease was about, bad humors or miasmas.
Right. Vague concepts.
Exactly. Then along came Rudolf Virchow in the 19th century. He's often called the father of modern pathology.
And he's the one who said,
He basically laid it out. Individuals are sick because their cells are sick. Simple, but revolutionary. It put the cell right at the center of everything. If your liver's failing, it's because the liver cells are failing.
Okay. So cells being sick, how does that start? What's the normal state?
Normal life for a cell is all about balance. Maintaining a steady state that's homeostasis. They operate in this really quite narrow comfort zone.
Like a thermostat, keeping the temperature just right.
Good analogy. And when things change, when stress hits either from outside or inside, the cell doesn't just give up immediately. Its first move is to try and adapt.
Adapt, so change its state to cope.
Precisely. Adaptation means making reversible changes to survive. Think about, say, muscle cells when you lift weights regularly.
They get bigger. Hypertrophy.
Exactly, that's adaptation. Or think about glandular tissue, maybe increasing the number of cells hyperplasia if there's more demand for hormones. The cell changes its structure or function to find a new steady state under stress.
But that adaptation has limits.
Oh, absolutely. There's only so much stress a cell can handle or adapt to. Our sources lay out this spectrum really clearly. Stress comes along.
And the cell either adapts,
Or, if the stress is too much, too fast, or just the wrong kind, it leads to cell injury.
Okay, injury? Is that the end?
Not necessarily, not at first. It starts as reversible injury. If you remove the stressor at this point, the cell can potentially recover, go back to normal.
But if you don't remove the stress,
Then it crosses a critical threshold. It goes from reversible to irreversible injury. And once that happens,
no turning back.
no turning back. Irreversible injury means cell death is inevitable. And that death happens mainly in one of two ways. Necrosis or apoptosis. We'll definitely dig into those.
Necrosis and apoptosis. Okay. But before we get there, there's something really surprising you mentioned from the sources about seeing the damage.
Ah, yes. The time lag. This is crucial and, frankly, a bit mind-bending.
What's the lag?
There's often a significant delay hours, even a day or more, between the moment a cell is biochemically doomed, meaning it's irreversibly injured.
So it's definitely going to die.
Right. It's past the point of no return. But the morphologic changes, the things you can actually see under a microscope, like the signs of necrosis, they take much longer to appear.
Wow. So the cell can be functionally dead, its fate sealed, but it still looks. Okay.
For a while, yes. Especially under a standard light microscope. The biochemical machinery might have completely collapsed minutes after a severe insult, but the structure holds together for hours.
That must make diagnosis tricky early on.
Extremely. Imagine a heart attack ischemia. The heart muscle cells might be irreversibly damaged within an hour or two. But if you took a biopsy then, they might not show the classic signs of necrosis yet.
So you can't just rely on looking at the tissue structure immediately after an event.
Not for an early diagnosis of irreversible injury, no. That's why clinicians rely heavily on biochemical markers, things leaking out of the damaged cells into the blood, like cardiac enzymes. Those markers tell you the damage is done long before the cell structure visibly disintegrates.
The biochemical leak is the smoking gun, even if the building looks intact.
Perfect analogy.
Yeah.
It highlights why understanding the molecular sequence of events is so vital. The visual evidence lags behind the functional reality.
Okay, that sets the scene. We have the timeline: homeostasis, adaptation, reversible injury, irreversible injury, death. Now let's get into the why. What actually causes cells to get injured in the first place? The etiology.
Right, the causes. And while there are countless specific triggers, they tend to fall into several major categories.
Where do we start? What's the most common culprit?
Without a doubt, number one is oxygen deprivation. Hypoxia. This is probably the single most important cause of cell injury and death worldwide.
Hypoxia, lack of oxygen. Why is that so catastrophic for a cell? More so than, say, getting physically hit.
Because oxygen is fundamental to energy production. It's the final electron acceptor in the mitochondrial electron transport chain.
The cell's power plant.
Exactly. Without oxygen, aerobic respiration shuts down. The cell can't make nearly enough ATP, the energy currency it needs for almost everything, maintaining ion gradients, synthesizing proteins, repairing damage.
So no oxygen, no power.
Pretty much. It cripples the cell's ability to function.
What kind of situations lead to hypoxia in the body?
Well, there are three main ways this happens. The most frequent is ischemia.
Ischemia. That means reduced blood flow, right? Like a blocked artery.
Precisely. Think heart attacks caused by blocked coronary arteries or strokes, often from blocked arteries in the brain. Ischemia is particularly nasty because it's not just lack of oxygen delivery.
What else?
It also means waste products aren't removed because the blood flow is cut off. So toxic metabolites build up, making the injury even worse than pure lack of oxygen alone.
Okay, so ischemia is number one. What else causes hypoxia?
Second is inadequate oxygenation of the blood itself, often due to cardiorespiratory failure. The heart isn't pumping well enough, or the lungs aren't exchanging gases properly, pneumonia, heart failure, things like that.
So the blood is flowing, but it's not carrying enough oxygen.
Right. And the third category is a decreased oxygen carrying capacity of the blood.
Like anemia.
Exactly. Severe anemia means fewer red blood cells, so less hemoglobin to carry oxygen. Or a really dramatic example is carbon monoxide poisoning.
Ah, yeah. The silent killer.
It is,
Yeah.
because CO binds to hemoglobin way more tightly than oxygen does, like 200 times more affinity. So even if you're breathing air with plenty of oxygen, the CO hogs all the seats on the hemoglobin bus. The tissues starve for oxygen, even though it's technically available.
And the cell's response to hypoxia depends on how bad it is.
Yes, mild chronic hypoxia might lead to adaptation.
Yeah.
The cell might shrink, atrophy to reduce its metabolic needs.
Yep.
But sudden, severe hypoxia, that causes injury and often rapid death. The speed and severity are key.
Okay, oxygen deprivation is huge.
Why?
What's next on the list of causes?
Next up are the physical agents. These are the more direct external insults.
Like trauma.
Yep. Mechanical trauma, getting hit, crushed, cut, which physically disrupts cells and tissues. Also, extremes in temperature.
Burns and frostbite.
Right. Intense heat denatures proteins and enzymes. Extreme cold can cause ice crystals to form inside cells, literally shredding them. Then you have things like radiation, electric shock, all causing direct physical or molecular damage.
Makes sense. Direct destruction. What about chemicals?
Chemical agents and drugs. This category is incredibly broad. It's not just about poisons like arsenic or cyanide, although those are definitely included.
What else falls under chemicals?
Even seemingly harmless substances in the wrong concentration can be injurious. Our sources point out high levels of glucose, like in uncontrolled diabetes.
Sugar. How does that hurt cells?
High glucose in the blood changes the osmotic balance. It can pull water out of cells or cause other metabolic stresses. Same with high concentrations of salt. It disrupts the cell's internal fluid and electrolyte environment.
So it's not always inherent toxicity. Sometimes it's just overwhelming the cell's balance.
Precisely. Context and concentration matter hugely. But then you do have the potent poisons. Cyanide, for instance, directly blocks cytochrome oxidase in mitochondria.
Mimicking hypoxia, essentially.
Yeah.
Cutting off the energy.
Exactly. And then add in environmental pollutants, air pollution, insecticides, industrial chemicals, alcohol, recreational drugs, even therapeutic drugs that have side effects or are taken in overdoses. It's a vast category because so many chemicals can interfere with cellular pathways.
Okay, physical, chemical.
Yeah.
What about living agents?
That brings us to biological agents. This covers the whole spectrum of infectious organisms.
Viruses, bacteria.
Fungi, parasites, even prions. They all cause injury in different ways. Viruses hijack the cell's machinery. Bacteria might release toxins that poison cells directly or indirectly. Parasites can physically invade and destroy tissues.
But it's not just the invaders, is it? Our own defenses can cause problems.
Absolutely, that falls under immunologic reactions. Sometimes the immune system goes awry.
Autoimmune diseases.
Like rheumatoid arthritis or lupus, where the immune system attacks the body's own healthy cells and tissues. Or sometimes, the immune response to an external agent like a virus or a transplanted organ is excessive or misdirected, causing collateral damage to normal cells. Inflammation itself, while protective, can be a major source of injury if it's uncontrolled.
The friendly fire of the immune system.
Oh, you could put it that way, yes.
Okay, we've covered external factors mostly. What about internal problems? Genetics.
Yes, genetic abnormalities are a huge source of cellular dysfunction and injury. These can range from subtle mutations to major chromosomal abnormalities.
How do gene defects lead to cell injury?
Many ways. A gene defect might mean a crucial enzyme isn't produced or it's non-functional. This leads to metabolic problems, like in lysosomal storage diseases, where waste products build up inside the cell because they can't be broken down,
Accumulating junk.
essentially. Or the genetic defect might cause proteins to misfold. The cell has systems to deal with misfolded proteins, but if they're produced too fast or the defect is too severe, they accumulate.
Why are misfolded proteins so bad?
They can aggregate, gum up the works. They trigger a cellular stress response called the unfolded protein response. If the cell can't clear the misfolded proteins, this stress response can actually push the cell towards apoptosis, programmed cell death, as a quality control measure. Damaged DNA from genetic defects can also trigger apoptosis.
A self-destruct sequence to prevent bigger problems.
In a way, yes. And the last major category of causes is nutritional imbalances.
Too little or too much.
Exactly. On one side, you have deficiency. Protein-calorie malnutrition is devastating globally. Lack of specific vitamins is also critical, as vitamins are often essential cofactors for enzymes. No vitamin, no enzyme function, cellular processes grind to a halt.
And the other side? Excess.
Yes, particularly relevant in developed countries. Nutritional excess. Obesity is a major driver of cell stress and injury, contributing to things like insulin resistance and inflammation. High levels of lipids, especially cholesterol, lead to atherosclerosis, where lipids accumulate in artery walls, causing injury and eventually ischemia. Too much of a good thing becomes toxic.
Okay, that's a comprehensive list of why cells get injured. Hypoxia, physical agents, chemicals, infections, immune reactions, genetics, nutrition. Now let's zoom back in on the process of injury itself, starting with that first stage, reversible injury.
Right. The point where the cell is showing signs of distress, it's functionally impaired, maybe structurally altered, but crucially, it can still recover if the damaging stimulus is removed.
The window of opportunity.
Exactly. And the most universal sign of reversible injury, especially in response to hypoxia or toxins that hit energy production, is cellular swelling.
Swelling. You mentioned this comes from ATP depletion. How does that work again?
It's quite direct. Cells pour a huge amount of energy ATP into maintaining ion gradients across their plasma membrane. The star player here is the sodium-potassium pump, Na plus K plus ATPase.
Pumping sodium out and potassium in.
Correct. It constantly pumps sodium out against its concentration gradient. This requires a lot of ATP. When ATP levels fall sharply, like in hypoxia,
The pump fails.
the pump fails or slows down drastically. Sodium that naturally leaks into the cell down its gradient isn't pumped back out effectively. So sodium accumulates inside the cell.
And water follows salt.
Always. Water moves passively by osmosis from an area of lower solute concentration, outside, to higher solute concentration, inside, now high in sodium. So water rushes into the cell.
Making it swell up.
Precisely. Generalized cellular swelling. This affects the whole cell, including organelles. The entire cell gets bigger, more turgid. If this happens to many cells in an organ, the organ itself becomes pale, heavier, and swollen.
And under the microscope, what does this swelling look like specifically?
Microscopically, you see what pathologists call hydropic change or vacuolar degeneration.
Hydropic, like water.
Exactly. You see these small, clear vacuoles appearing in the cytoplasm. These aren't empty spaces, though. They're actually segments of the endoplasmic reticulum, the ER, that have ballooned out with water and pinched off.
So the internal plumbing is swelling too.
Yes. The whole cell is taking on water, puffing up internally. This is a classic sign of reversible injury, particularly hypoxic or toxic injury.
Now, besides general swelling, the sources mention another specific type of reversible change, especially in certain organs. Fatty change. Steatosis.
Yes, steatosis is very common, especially in the liver, because the liver is central to fat metabolism. It can also happen in heart muscle, kidney, other organs.
What causes fat to build up reversibly?
Various injuries, particularly toxins like alcohol or metabolic conditions like obesity or diabetes, can disrupt the normal processing and export of triglycerides, the main type of fat.
So the fat comes in or is made but can't get out.
Essentially, yes. The metabolic pathways get overwhelmed or inhibited. So triglycerides accumulate within the cytoplasm, forming these lipid vacuoles. Initially, they might be small, but they can coalesce into large globules that push the cell nucleus right to the edge.
And this is still reversible.
In the early stages, yes. If you remove the cause, Stop the alcohol, manage the diabetes, the liver cells can process and clear the fat. But if it persists, fatty change signals significant metabolic dysfunction and can progress to more severe irreversible injury like inflammation, steatohepatitis, and scarring, fibrocirrhosis.
Okay, swelling, fatty change. And there was one more visual sign mentioned. The cell staining pinker, more eosinophilic.
Ah, yes, increased eosinophilia. This is also a very important indicator of cell injury.
Why does the color change?
Healthy cells have a lot of ribosomes,
Uh-huh.
rich in RNA, and intact nuclear chromatin. RNA binds the blue dye, hematoxylin, giving the healthy cytoplasm and nucleus a somewhat bluish tint, basophilia.
Okay, blue is healthy.
Generally speaking, yes. When a cell gets injured, a couple of things happen. First, ribosomes tend to detach from the ER and disperse, and RNA gets degraded, so the cell loses its blue staining capacity.
Left blue.
Second, proteins within the cytoplasm start to denature due to the altered internal environment, like lower pH from anaerobic metabolism. These denatured proteins bind more avidly to the pink red dye, eosin.
So lose the blue, gain the red?
Exactly. The net effect is that the injured cell cytoplasm appears much pinker or redder than normal. And this eosinophilia often becomes more pronounced as the cell progresses towards irreversible injury and necrosis. It's a visual marker of internal distress and protein damage.
So we have this picture of a reversibly injured cell. It's swollen, maybe filled with fat vacuoles, staining pinker, its pumps are failing, but it's still holding on. What tips it over the edge? What's the critical turn into irreversible injury?
That's the million-dollar question in pathology, defining that precise point of no return. It's often described as a somewhat nebulous threshold.
Not a clear on-off switch.
Not always biochemically sharp, but functionally, yes. It's the point where the damage becomes so severe that the cells simply cannot recover, even if the injurious stimulus is removed. The capacity for repair is overwhelmed.
What kind of damage pushes it past that point?
Two critical factors seem central. One, the inability to reverse mitochondrial dysfunction. If the mitochondria are severely damaged, ATP production cannot be restored.
The power stays off permanently.
Right. And two, profound disturbances in membrane function. This includes the plasma membrane losing its integrity completely and, critically, the rupture of lysosomal membranes.
Ah, the lysosomes containing digestive enzymes.
Exactly. When lysosomes rupture, those potent enzymes leak into the cytoplasm and start digesting the cell's own components. That's usually a fatal blow. Severe membrane damage overall is probably the key event marking irreversibility.
And this transition, this point of no return, it happens before we see the really obvious signs of death, like in the heart attack example.
Yes, let's revisit that timeline because it's so important. In myocardial ischemia,
Blocked coronary artery.
right, within minutes, you get ATP depletion, ion pump failure, cell swelling, that's reversible injury. But if the ischemia persists, say for maybe 30 minutes to an hour or two, depending on the cell type and conditions,
Then what?
the cell crosses into irreversible injury. Mitochondria are severely damaged, maybe calcium floods the cell, membranes start to fail. At this point, even if you restore blood flow right then, that cell is still going to die. Its fate is sealed.
But it doesn't look dead yet under the microscope.
Not necessarily showing the classic signs of necrosis, no. Those definitive changes, the nuclear breakdown, the intense eosinophilia, the fragmentation might not be clearly visible by light microscopy for another several hours, maybe 4 to 12 hours after the initial event.
So there's this window where the cell is doomed, but the microscopic evidence lags behind.
Precisely. This gap underscores why biochemical markers, like troponin leaking from dying heart cells, are so crucial for early diagnosis of things like heart attacks. They detect the membrane failure and leakage that signifies irreversible injury long before the cell looks fully necrotic.
Okay, so the cell has passed the point of no return. It's irreversibly injured. What happens next? The final step, cell death. You mentioned two main pathways.
Yes. Once irreversibly injured, the cell dies by one of two principal, morphologically and mechanistically distinct pathways: necrosis or apoptosis.
Necrosis versus apoptosis. Let's break them down. Start with necrosis. This sounds like the bad one.
Necrosis is generally considered the messy or accidental death. It's almost always a pathological process, the end result of severe irreversible injury from things like ischemia, toxins, infections, trauma.
What defines it? What goes wrong?
It's characterized by that severe mitochondrial damage we talked about, leading to catastrophic ATP depletion. And crucially, widespread uncontrolled membrane damage, both the outer plasma membrane and the internal lysosomal membranes rupture.
The lysosomes rupturing. that causes self-digestion.
Yes, the release of lysosomal enzymes into the cytoplasm leads to enzymatic digestion of cellular components. The cell basically dissolves from the inside out, losing all structural integrity.
Okay, let's look at the characteristics, like from table 2.1 in our source material. Cell size and necrosis.
The cell is enlarged. It swells up significantly due to the initial osmotic influx of water, and this swelling persists until it ruptures.
Membrane integrity.
Disrupted. The membranes lose their integrity completely, becoming leaky and eventually breaking apart.
And the cellular contents?
They undergo enzymatic digestion, as we said, and importantly, they leak out of the dying cell into the surrounding tissue.
That leakage must cause problems for the neighbors.
Big problems. The released intracellular contents, including specific molecules called damage-associated molecular patterns, or DAMPs, act as alarm signals. They trigger an acute inflammatory response in the adjacent tissue.
So necrosis always comes with inflammation?
Almost invariably, yes. The body sends in immune cells, like neutrophils, to clean up the debris from the exploded cells and deal with the cause of the injury. This inflammation is essential for healing, but can also cause further tissue damage itself.
Okay, and what happens to the nucleus during necrosis? There are specific stages, right?
Yes, the nuclear changes are hallmarks of necrosis. It typically proceeds through 3 stages, though you might not always see all 3 distinctly. First is pinosis.
Pinosis.
The nucleus shrinks down into a small, dense, intensely dark staining basophilic mass. The chromatin clumps together.
Shrunken and dark.
Then comes karyosis. The planotic nucleus fragments breaks up into smaller pieces.
Shattering.
Pretty much. And finally, karyolysis. The chromatin in those fragments is dissolved, likely by DNA's activity. The nucleus essentially fades away, loses its basophilic staining until it disappears completely. You're left with a dead cell, often just an eosinophilic ghost without a nucleus.
And the classic example of necrosis is?
Myocardial infarction, a heart attack, is the textbook example. A region of heart muscle dies due to prolonged ischemia.
And you can see this grossly.
Yes. If you take the heart out soon after a large MI and stain it with a special stain like triphenyl tetrazolium chloride, TTC.
What does that do?
TTC reacts with viable enzymes like dehydrogenases that are still present in living cells, turning the tissue magenta or red. But in the necrotic area, those enzymes have been denatured and lost. So the necrotic zone doesn't stain, appearing pale or yellowish white against the red viable tissue. It clearly delineates the dead area.
Okay, so necrosis is the messy, inflammatory death following severe injury. What's the alternative? Apoptosis.
Apoptosis is often called programmed cell death or cell suicide. It's fundamentally different from necrosis.
How so? Is it always pathological?
No, that's a key difference. Apoptosis is frequently a physiologic process. It's essential for normal development, like removing the webbing between fingers and toes in the embryo, or eliminating cells that are no longer needed during tissue turnover.
So it's a way the body gets rid of unwanted or potentially harmful cells cleanly.
Exactly. It's also used to eliminate cells with damaged DNA that can't be repaired, preventing cancer, or cells infected with viruses, or self-reactive immune cells. So it can be pathological too, but it often serves a protective or developmental role.
And the mechanism is different, not just membrane rupture.
Totally different.
Yeah.
Apoptosis is a highly regulated, energy-dependent process involving the activation of specific enzymes called caspase.
Right.
These caspases orchestrate the controlled dismantling of the cell.
Dismantling, not exploding.
Right. Think of it as neat demolition versus uncontrolled explosion. Let's look at the characteristics again. Cell size and apoptosis.
It's reduced. The cell shrinks as its cytoskeleton is broken down and the cytoplasm condenses.
Membrane integrity.
Remarkably, the plasma membrane remains intact throughout the process. However, its structure changes subtly. Lipids, like phosphatidylserine, normally on the inner leaflet, flip to the outside. Why does it do that?
That lipid flip acts as an eat-me signal for phagocytic cells like macrophages.
Clever. And the cellular contents?
They are neatly packaged. The cell breaks up into membrane-bound fragments called apoptotic bodies. These contain portions of the cytoplasm and organelles, and often fragments of the nucleus.
So nothing leaks out?
Correct. Because the contents are contained within these sealed vesicles, they don't spill into the surrounding tissue.
And the consequence of no leakage?
No inflammation. The apoptotic bodies are quickly recognized and engulfed by phagocytes before they can rupture. The whole process is typically silent, causing minimal disruption to the surrounding tissue.
Very tidy. What about the nucleus in apoptosis? Does it go through pinosis, karyorexis, karyolysis?
The nuclear changes are also distinct. The chromatin condenses, often aggregating peripherally under the nuclear membrane. But the key feature is fragmentation of the nucleus into nucleosome-sized pieces. This is done by specific endonucleuses activated during apoptosis. It's a very orderly breakdown, unlike the chaotic dissolution in necrosis.
So necrosis is murder, messy, inflammatory. Apoptosis is suicide, clean, non-inflammatory.
That's a great way to summarize the core difference, yes. Understanding which pathway is involved is crucial for understanding the disease process.
Okay, we've seen the differences under the light microscope. Let's go deeper. To the electron microscope level, what ultrastructural evidence confirms these injury processes? What details emerge at that magnification?
Electron microscopy, EM, gives us a much finer view of the damage, especially early on. In reversible injury, even before major light microscopic changes, EM shows distinct alterations.
Like what?
The plasma membrane starts showing blebbing little bulges popping out, plus blunting or loss of microvilli. This reflects cytoskeletal disruption and the struggle to contain the osmotic swelling.
So the cell surface looks stressed. What about inside, or mitochondria?
Mitochondria show changes very early. They swell significantly again due to water influx and ion shifts. But critically, even in reversible injury, you might start seeing small, amorphous densities appearing within the mitochondrial matrix.
What are those densities?
Their exact composition is debated, maybe proteins or lipids, but their appearance is considered an early sign that things are heading towards irreversible damage. They indicate mitochondrial dysfunction.
And the endoplasmic reticulum? We mentioned it balloons out into vacuoles.
Yes, EM confirms this ER dilation is massive. You could also see the ribosomes, the little dots responsible for protein synthesis, detaching from the ER surface, detachment of polysomes. This visually confirms that protein synthesis is shutting down.
A sign of functional collapse.
Absolutely. And EM reveals another fascinating feature of damaged cells. Myelin figures.
Myelin, like nerve insulation.
Same name, different context. These are world-laminated structures like onion layers composed of phospholipids. They are derived from damaged cellular membranes, plasma membrane, mitochondrial membranes, ER membranes. As these membranes break down, the lipids aggregate into these characteristic figures.
So they're like the wreckage of the membranes piling up.
Exactly. They are ultrastructural debris, a clear sign of membrane damage, visible in both reversible and irreversible injury, but more prominent as the cell degrades.
How does the EM view help distinguish the path to necrosis versus apoptosis?
It really emphasizes the mitochondrial role. In necrosis, the EM picture is dominated by severe mitochondrial swelling, disruption of their cristae, internal folds, and large amorphous densities, reflecting complete energy failure and membrane damage.
Total meltdown.
Right. In apoptosis, while mitochondria are involved in triggering the process, releasing things like cytochrome C, they often don't show that same degree of massive swelling and disintegration early on. The apoptotic cell maintains more structural orderliness during its programmed disassembly, which EM clearly shows.
Okay, EM confirms the internal chaos of necrosis versus the controlled breakdown of apoptosis. You also mentioned reactive oxygen species playing a role.
Yes, ROS, reactive oxygen species. These are chemically reactive molecules containing oxygen like superoxide radicals O2, hydrogen peroxide HCO2, and hydroxyl radicals.
Radicals.
Essentially, yes. They are generated normally in small amounts during mitochondrial respiration, but cells have antioxidant defenses to neutralize them.
But in injury.
In certain injury states, especially ischemia reperfusion injury, where blood flow is restored after a period of blockage, or exposure to toxins or radiation, ROS production can skyrocket, overwhelming the defenses.
And what do these ROS do?
They're incredibly damaging. They attack lipids in membranes, lipid peroxidation, causing them to break down. They modify proteins, causing them to lose function or misfold. And they can directly damage DNA. This widespread oxidative stress is a major contributor to pushing a cell from reversible to irreversible injury, particularly necrosis.
So the return of oxygen can sometimes be the final blow.
Paradoxically, yes. Reintroducing oxygen to severely ischemic tissue can trigger a burst of ROS production from damaged mitochondria, leading to massive secondary damage. It's a critical factor in the pathology.
All right, before you wrap up, let's touch on a couple of other cellular responses or fates related to injury. Autophagy was mentioned.
Autophagy meaning self-eating. This is a fascinating survival mechanism.
How does it work?
When a cell is stressed, particularly by nutrient deprivation, it can activate autophagy. It essentially creates double-membraned vesicles called autophagosomes that engulf bulk cytoplasm, proteins, or even entire damaged organelles like mitochondria.
And then what?
These autophagosomes then fuse with lysosomes, and the contents are digested. The breakdown products, amino acids, fatty acids, are recycled back into the cytosol to provide nutrients and energy.
So it's a way to survive lean times by recycling internal components.
Exactly. It's primarily adaptive. It helps clear out damaged components too. However, if the stress is too severe or prolonged, autophagy can become excessive and contribute to cell death itself, sometimes called type 2 programmed cell death. Its role is complex, both pro-survival and potentially pro-death.
And lastly, the long-term consequences, accumulations and calcification.
Right. These aren't death pathways, but common results of chronic injury or metabolic problems. Intracellular accumulations are just what they sound like. Buildup of substances within the cell.
Like the fat and steatosis.
Yes, lipids are common. But also abnormal proteins, like in neurodegenerative diseases, glycogen and glycogen storage diseases, pigments, like hemosiderin from excess iron, or carbon dust in smokers' lungs. These accumulations happen when the cell produces too much of something or can't metabolize or transport it properly. They impair function.
And calcification. Hardening.
Pathologic calcification is the abnormal deposition of calcium salts, usually calcium phosphate, in tissues. There are two main types.
Which are?
Dystrophic calcification. This occurs locally in areas of dying or necrotic tissue, even if blood calcium levels are normal. It's like a tombstone marking previous injury. You see it in old scars, damaged heart valves, atherosclerotic plaques.
So what happens where tissue is already damaged?
Right. The second type is metastatic calcification. This occurs in normal, viable tissues, but is caused by systemic hypercalcemia, too much calcium in the blood. This might happen due to kidney failure, certain tumors, or vitamin D disorders. The excess calcium deposits widely, for example, in the lungs, kidneys, stomach lining.
Okay, that makes sense. Dystrophic is local in dead tissue. Metastatic is widespread due to high blood calcium.
Precisely.
Wow. We've covered a huge amount of ground, from the first adaptation to the final deposition of calcium. Let's try to synthesize this. What are the absolute key takeaways for someone listening?
I think the first is grasping that fundamental split in cell death. Necrosis versus apoptosis.
Right. The messy versus the clean.
Necrosis. Pathological, swelling, membrane rupture, enzymatic leakage always triggers inflammation. Think trauma, ischemia.
Apoptosis. Often physiological, shrinking, intact membranes, packaging into apoptotic bodies, no inflammation, silent removal. Think development, eliminating damaged cells.
Understanding that difference is job number one.
What else?
The concept of pathogenesis. It's not just knowing the cause, etiology, but understanding the sequence of events. How does hypoxia lead to ATP depletion? How does that cause pump failure and swelling? How does that lead to irreversible membrane damage and necrosis? Tracing that molecular and cellular cascade is what pathology is all about.
So the how is just as important as the why.
Absolutely, because interventions often target steps in that sequence. And finally, don't discount morphology.
The visual changes. Even with all the omics, genomics, proteomics.
Even then, seeing the hydropic change, the eosinophilia, the nuclear pinosis, the apoptotic bodies under the microscope that remains a cornerstone of diagnosis, it tells you what is happening and where. Molecular tests often refine the diagnosis, but morphology provides the crucial context.
Okay, so we started by talking about adaptation, cells trying to cope, the heart muscle getting bigger, hypertrophy, under high blood pressure.
Yes, a classic adaptive response. The cell gets bigger to handle the increased workload. It survives for a time.
Which leads us perfectly into our final provocative thought for you, the listener. That adaptation, that hypertrophy, it's a survival mechanism, right?
Initially, yes.
But think about that bigger muscle cell. It needs more oxygen, more nutrients, a bigger blood supply than the original cell. By adapting to one stress, high pressure, it has made itself inherently more metabolically demanding.
It has increased its baseline needs.
So here's the thought. What happens when that adapted, hypertrophied heart faces a new stress, like a slight reduction in coronary bleed flow that a normal heart might tolerate?
That previously successful adaptation now becomes a major vulnerability. The cell that grew to survive is now the first to die when supplies get tight, precisely because it grew.
So how often is the ultimate failure in disease not directly due to the initial insult, but actually the long-term consequence, the hidden cost of the cell's very attempt to adapt and survive? When does success pave the way for failure? Something to think about.